I hope we've made it fairly clear that Darwin's theory really doesn't account for the molecular beginnings of life. Is there anybody question that? If you've been with us since day one, if you've been watching the YouTube videos, if you've been watching online, we've made it pretty clear that Darwin's theory doesn't really account for the molecular structures of life. And if the fossil record and if the homology record has not proven that, we're going to really prove it tonight. We're going to really prove it tonight, because there just is no good evolutionary explanation behind some of the things we're going to look at tonight. And the explanations that are attempted to be given are, let's just say, weak at best and silly at worst, even by educated men with PhDs and so forth in biochemistry, because they want to continue to cling to the evolutionary idea. That's the only reason. They have to come up with an explanation, and they really have trouble coming up with some of the stuff we're going to look at tonight, an explanation for some of the things we're going to look at tonight, such as the clotting cascade and vision and things like that. And the point of this, we're going to go through that stuff when we get to it really pretty quickly. And the reason we're going to go through it quickly is because it's not the intent to make you scientists in this class and for you to memorize the clotting cascade. Trust me, as a medical student, I had to do that. And it's no fun, and you don't remember it. I forgot it already, and I got to look at it again now. So that's not the point. The point is to show the complexity of it and try to figure out, if you're a scientist, how these things could have possibly had come about from an evolutionary point of view. So Darwin's theory just does not account then for the molecular structure of life. What are we talking about in essence here? Early in the 19th century, two scientists, Matthias Schleiden and Theodor Schwann, put forth the cellular theory of life. Do you know what that means when we say the cellular theory of life? I mean, all of you here that have had biology, you know that all of us are made of cells. Every life form we have is made of cells. Now, some life forms are singular cells, like bacteria. Bacteria is a single cell. It doesn't need more than one. Most bacteria, some bacteria are in chains, whatever. But they're cells. But when you get into higher life forms, you get into plants and you get into animals, you get into human beings, really what we are is a collection of trillions of cells. And that theory was first postulated in the 17th century by these two scientists. What's in the cell? All kinds of things. And we're not going to study all this tonight, but there's the eukaryotic and prokaryotic cells, and we're eukaryotic cells. Our bodies are made of eukaryotic cells. And eukaryotic cells have a nucleus. They have Golgi bodies. They have other organelles like the centromere and so forth that are in it. And they all have a purpose. And they all had to come, if you're talking about it from an evolutionary point of view, each one of those organelles had to have an evolutionary beginning, didn't it? I mean, if you're trying to explain life, then not only do you have to explain, where the bacteria came from, but you got to start explaining where some of these organelles became functional. How did that happen from an evolutionary point of view? I mean, you have to do that if you're an evolutionist. 19th century scientists knew about the cell. They were able to, with their microscopes, they could see the cell, they could see the nucleus, they could see the organelles where they're inside the nucleus. So they had an understanding by the end of the 19th century, and certainly during Darwin's time, what cellular structure was. And as we've already said, they had proposed the cellular structure of life. I still find it amazing, don't you, that we are made of trillions of cells. But which cell is me? You're made, all of us have trillions of cells, but which one is you? I don't know. I don't have the answer to that. It's integrated, and we're all made of one cells. And you can take some of my cells away, but I'm still here. You can take a lot of my cells away, but that's a different topic altogether. But I just find it interesting. But somewhere along the line of our development, we all began as one cell. That's how we began. That one cell divided. It was fertilized. You had two different cells coming together to form one cell. And then it became 2 cells, and then 4, and then on and on and on until we're now trillions of cells. And somewhere along that line, all those cells had to differentiate between bone and brain and muscle and intestine and heart, and yet we're all here. The 19th century scientists, they sort of knew that, and they knew the structures. But you know what they didn't know? They didn't know how those structures function, and they certainly did not know the chemical aspects of those organelles. They didn't have the chemistry behind it. And so, as Michael Behe put it, for Darwin, the cell was a black box. You know what a black box is? I mean, you know what a black box is, but what the concept of a black box is. This is a black box for me, this computer right here. It's a black box for me. Now, for some of you, it's probably not. But for me, that's a black box. I mean, I know what it does, but it would be impossible for me to explain that. Same thing with my cellular phone. And I know that I receive calls and I can get pictures on it now, and that I can send messages, and that totally amazes me that we can do that. And it's a black box for me because I don't know how it works. Well, for Darwin, the cell was a black box, and it was for all the 18th, 19th century scientists and at the beginning of the 20th century. And so in 1996, Michael Behe published Darwin's Black Box, which introduced the concept of irreducible complexity. And that's a fancy word, but it's really a very easy concept to understand. And the point is, Darwin could not have explained how those cells operated because they didn't have the biochemistry then. So it was a complete mystery for him. The question Behe asked, and by the way, that book, how many have read Darwin's Black Box? Has anybody read it? One? You haven't read it. If you're really interested in this kind of stuff, I would call it a classic. And my son Toby actually introduced me to this years ago. He brought it home from college, as I recall. I looked at it and I'm going, what is this? So I just started reading it. And it's a classic. I would put it in the classic in the same line as Jonathan Wells' Icons of Evolution, as well as Michael Denton's Evolution Theory and Crisis, those books, and some of Stephen Meyer's. His is definitely a classic. 1996 he wrote it. But what he asked was, how could these biochemical systems be gradually produced? Because they couldn't just have sprung forward. That's not evolution. You can't just have, some of these organelles and some of these cellular functions just spring up. It's one thing to propose a mechanism for gradual changes that must occur morphologically or structurally, and it's a whole different thing to describe biochemical gradualism, which is actually even more necessary. So you got to do more than describe how some flies can develop 4 wings instead of 2, even though they're non-functional. You got to start explaining other things as well. So it's one thing to say structural, that how we look at the fossils and how those structures change. It's quite another to explain the biochemistry. And yet Darwin's theory demands gradualism. Darwin demands gradualism. You will see this quote. I've already quoted it once in this class, if you remember, right at the very beginning. You'll see this quote again before this class is over, because it is part of the evolutionary theory. It is the dogma of evolutionary theory. And Darwin said in his book on the origin of species, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. And if Behe's book, Darwin's Black Box, doesn't show Darwin or neo-Darwin proponents that it does break down, then nothing does. It's just that simple. So are there systems that could not be formed by numerous successive slight modifications? What do you think? Well, there's many systems, many systems, most systems for that matter, but many systems that cannot be explained from numerous successive slight modifications. So, what is the concept of irreducible complexity? What do we mean by that? And for this, I'm going to quote Michael Behe. This comes from Darwin's Black Box. By irreducibly complex, I, meaning Michael Behe, mean a single system of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. So you've got a multiple series of parts or function of either an organ or a chemical reaction that must occur, and if you remove any one of those parts, then the system will fail and is non-functional. So it's really not difficult at all to understand the concept of irreducible complexity. So Darwin proposed numerous small changes must occur anatomically for a change in the organism to occur. And that's what we're looking at. Well, this may have some logic. I don't agree with it, but it may have some logic when you investigate things like vision on the chemical level. Darwin's theory fails on even a grander scale, on an even bigger scale than it does morphologically. Now, Behe in his book used a mousetrap, and if you were in my class a few years ago, y'all remember I actually brought a mousetrap up here. Well, I brought a picture today. I couldn't find a mousetrap, but in my house. I'm sure I could have bought one, but this is a mousetrap. Pretty simple thing, not really a complicated thing. What's the purpose of the mousetrap? Well, it is to capture a mouse. That's putting it kind of the euphemistic way. It is to capture, or as Michael Behe puts it, it is to immobilize a mouse. That is the purpose of your basic mousetrap. And so it has several structures. It has a spring, it has a hammer, it has a platform, it has a holding bar, about 5 different things that it has. And if you were trying to evolve a mousetrap, how and why would any one of these components develop if they had no function in and of themselves? So for example, if all you had, you're trying to immobilize a mouse, that's your job, that's what you're trying to get to. And so you have a spring, but you don't have a hammer, you don't have a platform, you don't have any of these other things. Is that spring going to do you any good at all in killing that mouse? No, it's not. Okay, I got the hammer. If I have, all I have is the hammer. That's the part that comes down, I guess. That's what they call it. And that's all I've got. I don't have the spring. I don't have the holding bar. Don't have the platform. Is that going to help me immobilize a mouse? No, it's not. Point is, you need all 5 of those things. You need every one of those. So the question is, how do you evolve to the point where you get a mousetrap when none of these other 5 things had any function until they all came together to form a mousetrap? That's irreducible complexity. That's what it is. You've got, if you're a scientist, if you're an evolutionist, you've got to somehow explain how in the world you get those 5 components coming together, which with each component having to evolve somehow on its own. And so it's not a difficult thing at all. Now, when you see the film in 2 weeks, you will see they'll be looking at the flagella. I didn't include the flagella in your handouts. The reason is I knew that the video was going to do a much better job explaining the flagella. You know what a flagella is? A little thing that bacteria have that allows it to move around. It whips around all over the place, and the bacteria is able to move around. It turns out people have spent decades of their life studying the flagella and how the flagella works. And it comes down that there's actually 40 different components to a simple flagella. And you've got to explain where those components came from. So they will do that next week. Or we will look at that next week. We'll look at it for 2 weeks, excuse me. But as Michael Behe says, irreducibly complex systems are nasty roadblocks for Darwinian evolution. And they need minimal function. The need for minimal function greatly exacerbates the dilemma. And what does he mean by minimal function? He means for one of these things to develop, they've got to have some function, or there's no reason to develop them. And so minimal function is a necessity, and it makes it even harder for Darwinian evolution. Many irreducible systems, including cilia, flagella, the antigen antibody and complement cascade, the bombardier beetle, you remember that from a few years if you were in this class? We're not going to look at that either, but that's in Darwin's black box, and the clotting cascade, and we could add many, 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 many more to that. Here's a way of thinking about it. Now, Behe in his book talks about Rube Goldberg. Anybody remember or know who Rube Goldberg is? Okay, you know his work, but you don't know his name if you're probably at least 40 years old or older probably, because he used to have these cartoons that you would see in magazines and papers and stuff like that. And this happens to be, and I know it's hard to see, so I apologize for that. Probably will look better on the YouTube than it does here. But this is actually a pencil sharpening machine. That's what that is. Now, the deal is you go through all these various pulleys and wires and steps and water falling and buckets and all that till eventually you get the pencil sharpened at the end. And that's a silly thing. We go, that's silliness. But in actuality, if you removed any one of those parts, the system fails, doesn't it? So that's what you got to think of when you think of irreducible complexity. If you remove any one of those, you could almost sort of liken it to dominoes in a way. You remove a couple of dominoes and you're trying to turn those dominoes over, you're going to stop. And that's what's going to happen. It's going to function to a point and then it's going to stop. So think of this when you're thinking of irreducible complexity. Modern biochemists have discovered many Rube Goldberg systems. They have. We're going to look at a few. Like I said, I'm going to go through this pretty quick. This is in your handout, and again, the point is not to make scientists of us all, but just to show a few systems that prove or show irreducible complexity. And the first thing we're going to look at is vision, the eye. The anatomy of the eye was known by Darwin. In fact, Darwin, in his book on Origin of Species, tried to explain how the eye could have come about from an evolutionary process. He was looking at it anatomically, okay? He looked at the lens and he looked at the pupil, which acts as a shutter. The lens gathers light and focuses it. You got the retina, where the forms the shape and the image, and the muscles of the eye, which allow quick movement, all make up the modern mammalian eye, and even the non-mammalian eye for that matter, for the most part. And we knew that. So modern man still cannot explain everything about vision biochemically, but we have at least begun to approach an understanding of it. And so what follows is sort of our best explanation of how vision works to date, and it is not a complete explanation. Neither is the clotting cascade when I get to it. There's a lot more involved even than what's here. But this gives you at least a feel of how complex it is. And so again, we're going to run through this pretty good. This is how you're able to see. This is how vision works. When light hits the retina, a photon interacts with a molecule called 11-cis-retinol, rearranges instantly in picoseconds, that's real short, to form trans-retinal. This change in trans-retinal forces a change in the shape of the protein called rhodopsin, which then metamorphosizes into meta-rhodopsin 2, sticks to a protein called transducin. Before said transducin had been tightly bound to GDP. GDP then falls off, and GDP now binds with this transducin. This GTP transducin meta-rhodopsin now binds with a protein, phosphodiesterase, in the membrane of the cells, with this attachment, phosphodiesterase cuts a molecule called cyclic GMP. Phosphodiesterase lowers cyclic GMP's concentration. Then when cyclic GMP is reduced, an ion channel closes, causing a positively charged sodium ion to be reduced. This imbalance in the charge causes a current to be transmitted down the optic nerve to your brain, resulting in vision. Did you get all that? So that's irreducibly complex. There's a bonus question. If somebody can tell me this word phosphodiesterase, what is a common phosphodiesterase inhibitor in medicine? Viagra. So the phosphodiesterase has other functions too, but that's vision. Okay, now you got a problem. Okay, so the question then is how did all these chemicals come into being, and what function did they have before vision was accomplished, and what would be the purpose for said function? What would have been all those chemical reactions going on if they didn't ultimately result in vision, right? That's irreducible complexity. That's not the only problem you have though, and I've got it in your book, but once you have vision, then you have to have everything converting back to its original form, or for this whole process to reoccur again in picoseconds. We're not going to go into that tonight. So as Behe puts it, anatomy is quite simply irrelevant to the question of whether evolution could take place on the molecular level. It's just irrelevant. If you can't explain it biochemically, then anatomy becomes even irrelevant to a point, right? You haven't even begun to explain it biochemically. Okay, the clotting cascade. Like I said, when I was in medical school in the dark ages, yes, they knew how the clotting cascade occurred. They know more about it now than they did 30 years ago, I'm sure. But this was the bane of our existence, memorizing stuff like you're going to find today in medical school, memorizing the clotting cascade. Now, when you look at the clotting cascade, I want to explain a few things, because sometimes even in medical school, they didn't do a good job making it easy on you, trying to memorize this stuff, because you get a lot of names, you get a lot of factors, you got a lot of numbers, you got zigzags, you got everything going on, and you got intrinsic pathway, extrinsic pathway, and it gets a little confusing. But just know this: there are at least 13 clotting factors. They're numbered with Roman numerals. Why Roman numerals? I don't know, to make it difficult on us. But that's how they're done. Roman numeral 1, 2, 3, all the way down through 13. There's actually more than 13, but those are the ones that they have the numbers attached to them. Some of these have their own name. In fact, they've all got their own name except factor 5. Factor 5 still doesn't have a name. It's kind of, we're figuring out a name. I don't know, it doesn't have one. So when you see these clotting cascades, sometimes you'll see them by name, sometimes you'll see them by number. What I've tried to do for you, with the help of my lovely wife, Cherry, because I wrote this out, and if I would have printed this out and given it to you, you couldn't have read it. So she printed it out for me. I tried to put, if you've got it in front of you, I've tried to put the numbers with the name. For example, and it goes from the top down. Clotting factor 13 and 12 is where it all begins. Now you'll see on this that there's not 13 in a row. It's not that simple. But I've tried to simplify this for you as much as I can. So you'll see the clotting cascade. But it starts with the wound. First of all, what would happen if you had, I mean, you know what would happen, but let's say I put a carton of milk up here and I poked a hole in that milk. What's going to happen? I mean, the milk's going to all drain out, and you're going to have an empty carton in a few minutes, right? Or a few seconds. Well, fortunately, our bodies aren't like that. If I were to poke a hole in my finger, do you think I would bleed to death? When you go to the doctor and you give blood, or you go to the lab and you get blood, do you bleed to death? Well, you might if you had a severe bleeding disorder, but you probably wouldn't have made it to that point if you had that bleeding disorder. So the body is made so that we don't bleed to death when you get a simple cut. Now, a big enough cut, your body can't clot fast enough. But obviously, small cuts, you're going to be able to survive minor cuts and scrapes because your body clots. Clotting is very, very, very important. I don't guess I need to tell you that, but it's a very important thing for life. Too much clotting is not a good thing, okay? So the body has to turn that clotting mechanism off, or if it didn't, what would happen to you? Before long, your whole arteries would be plugged up with clot, and you would be dead. So if it's a good thing for your body to clot, it's a good thing that your body don't keep clotting once that cascade is started. So this cascade gets started, it has to be turned off just as rapidly as well, or we're in big trouble. And a lot of the medications that you know people are taking today, yours truly included, and some of you in this room probably, take medications to keep their body from clotting so quickly. Because if they didn't, they would have strokes or heart attacks. So it's a very, very obviously important fact of life that your body clots and that your body is able to stop the clot. And this is how it works, one more time, in a kind of a quick fashion. Thrombin initially exists as prothrombin, and I'm going to give you the quick version and the long version. Thrombin initially exists as prothrombin. Prothrombin must be activated by Stuart Factor, which is also factor 10. Stuart Factor cleaves thrombin to activate thrombin to cleave fibrinogen to form fibrin to form the clot. All clots are made of fibrin. You've heard of a fibrin or fibrous clot. And you look at it underneath a microscope, and it looks like little strands coming together to form that fibrous clot. This is the quick version. That's not the cascade, actually. That's the quick version. Now we're going to go through the quicker version. Accelerin is needed to increase the activity of Stuart Factor. Accelerin exists in the inactive form proaccelerin. What you'll see about the clotting cascade is every one of these clotting factors has an inactive form and an active form, and they're all activated by the factor that precedes it once it becomes activated. Hence the name cascade. So proaccelerin, which in turn is activated by thrombin, there's always a bit of thrombin in your bloodstream. It's there circulating, but it has to be triggered. So back up a little bit. Prothrombin, before it can be turned on into thrombin, must be modified by glutamate residues change to gamma carboxyglutamate. For this to occur, vitamin K must be present. Most of you know if you don't have vitamin K, what's wrong? You're going to bleed. One of the drugs, this is just an aside, it's not part of the evolution, but I thought it was interesting. Ever heard of Coumadin? Also known as warfarin. Warfarin is actually derived, that name, from Wisconsin Alumni Research Fund. I guess they discovered it. What does it do? It looks like vitamin K, but it's not vitamin K. So what that does to you is it makes your blood bleed easier, makes it, patients like to use the word thinner, but it keeps you from clotting so quickly so you don't have a blood clot that goes to your lung or your brain. That's what vitamin K. And by the way, not in your list, but calcium is also a clotting factor. If some of these reactions, not all of them, but some of these reactions could not occur if you didn't have calcium in your bloodstream as well. In its presence, prothrombin cannot be active, which slows the clotting mechanism down. Stuart Factor is activated by intrinsic and extrinsic pathways. And for a medical student, this is what really gets confusing. What is an extrinsic and intrinsic pathway? Well, your intrinsic and extrinsic pathways both are part of you being able to clot. To a large extent, this is a test tube problem. If you took blood out through the vein and you let it sit, there's no injury in that test tube, but what happens to your blood if you let it sit? It clots. So that is the intrinsic pathway. For the extrinsic pathway to occur, a wound must occur, or else again, you'd be clotting all over the place and you'd be in big trouble. So the difference between intrinsic and extrinsic is really more about a laboratory test than anything else. There's a lot of crossover. With the intrinsic pathway, all the proteins for clotting are in the plasma. The extrinsic pathway, some clotting proteins occur on the cells. And so let's look at intrinsic. I'm trying to make this as confusing for you as I can so you can see what irreducibly complex means, okay? An animal receives a cut, and as a result, a protein called the Hageman factor sticks to the cell surface near the wound. Bound Hageman factor is cleaved by HMK protein and yields activated Hageman. Hageman converts to prekallikrein to kallikrein, which speeds up the conversion of more activated Hageman. Hageman and HMK transfer PTA to activated PTA, which activates convertin, which activates the Christmas factor, which is also a clotting factor, which has an actual name to it. Christmas factor with anti-hemophilic factor changes Stuart factor to its active form. With the extrinsic pathway, proconvertin becomes convertin by activated Hageman and thrombin with tissue factor. Convertin changes Stuart factor to its active form. Tissue factor appears only outside the cell and is usually not interact with the blood. Only when injury brings tissue factor into contact with the blood is the intrinsic pathway initiated. Intrinsic and extrinsic pathways then cross. So, it is obvious, it's an obvious key that clotting cascade has to be turned off. Once you turn it on, you better be able to turn it off. And if you didn't turn it off, you were in big trouble. So you have to keep that in mind. Also keep in mind that for the clotting cascade, none of the components of the system are used for anything but controlling the function of the clot. That's what they're there for. That's what the Hageman, I don't know of any other function of Stuart factor than this clotting cascade. I don't know of any other function for Hageman. Now, calcium's got other functions, but we're talking about these other specific clotting factors. That's all they're involved in. There's no other function in life. So what stops the clot? Kind of reverse everything. Antithrombin binds active clotting proteins. These bind to heparin, found inside the cells of undamaged blood vessels. You even know what heparin is. Heparin's what they give you when you go in the hospital in the IV form or in a shot form. Then they give you warfarin before you leave, okay? Because nobody wants to shoot themselves when they're leaving the hospital. A protein known as protein C then destroys Accelerin and activates anti-hemophilic factor. Thrombomodulin then binds with thrombin, and the clot is stabilized by fibrin stabilizing factor. Eventually, the clot is removed by plasma, activated into its inactive form, plasminogen, by a protein called TPA. You know what TPA is, and the medical people know what TPA is. If you go into the hospital tonight with a stroke that's occurring and it's a clot, it's not a hemorrhage, what are they going to give you? TPA, because they're going to break that clot up. So a lot of our medications come from this. Another protein called alpha antitrypsin also controls the clot dissolution by binding with the fibrin clot. In addition to all that, there's a few other ways that your body helps control bleeding. And one is contraction of the blood vessels themselves. Give someone epinephrine. What do we use epinephrine for when we're given a local anesthetic? Keep the bleeding down because it causes blood vessels to constrict. And also, also you have platelets. We didn't even talk about platelets, but platelets are necessary as well for the clotting mechanism to occur. They cause the stickiness of the clot. So I'm not going to go over that. We just did it, but that's for you to look at. But it shows up better in your handout probably than it does on that screen. But my point in the clotting thing is, how do you get that by evolution? The point of clotting is for the organism to survive, correct? Or else if you got a minor cut, you would die. If you didn't have all of these, what's hemophilia? Hemophilia, people don't survive very long with hemophilia, do they? That's one of these factors not working, the Christmas factor, or maybe the Stuart factor. There's different forms of hemophilia. So in order for the survival of the organism, you've got to get to the bottom. And if you took out any one of these, you got trouble. Now, have evolutionists tried to give an evolutionary explanation for the clotting factor? They have tried. And he talks about this in his book. And he refers to a guy by the name of Russell Doolittle, who's a professor of biochemistry at the University of California, San Diego. And I read his explanation in the book, Darwin's Black Box. And it's, if the guy wasn't a PhD and well-renowned biochemist, you would think, are you kidding me? When you read it. He offers no causation for the various clotting factors, and frequently throughout his explanation are words such as appears, Hageman factor, appears, springs forth, something springs forth. These factors are born. They're born. Or they are unleashed. Does that sound scientific? Does that sound like a good explanation to you? That's what they're offering. Bottom line is, when you look at, Behe's been criticized by chemists and everything else, because you're a creationist, Behe, you're a creationist. But you know what? They've never given a good explanation. Give me a good evolutionary explanation instead of using words like it appears. That's like saying, how do you become a millionaire? Well, first give me $1 million. You know, I can become a millionaire real quick. So in Doolittle's account, clotting does not occur till the last stage. And this begs the question. How do you get from one step to the other without the organism bleeding to death first? How do you get there? How do you get from that last step? And how do you get from each one of these steps where death hasn't occurred before it occurs? That's our 5-minute warning, so I got time to... I'm going to go through this very quickly. The endocrine system. Pituitary gland inside your body, master gland of the body, produces at least 8 hormones. It controls growth, blood pressure, some aspects of pregnancy, the production of breast milk, control of our sexual organs, metabolism, temperature, osmolarity, and thyroid functions. You would be dead without a pituitary gland. How did the body come up with that gland to control 8 different functions? How did evolution describe that? I'm just going to describe one, the thyroid. Thyroid produces thyroid hormones which control metabolism of our bodies. If you don't have enough thyroid, you're sluggish, you're gaining weight, your hair's falling out, you feel cold all the time. If you have too much thyroid, you're losing weight, you're hot all the time, you're sweating easily, and your body and your heart's racing with you. Your thyroid has to be very narrowly controlled, and when there's a problem, you know it. The thyroid gland must receive information from the pituitary gland. It couldn't work if it didn't have information coming from it from the pituitary gland. And guess what? The pituitary gland receives information from the hypothalamus. That's quick, I know, but I wanted just to explain that one to me, not the rest of the pituitary functions, which we really don't have time to go into. Final thoughts. Evolution has no answer to irreducible complexity. It just does not. There is no good explanation that I've seen. Have they tried? Oh yeah, and Dr. Doolittle's not the only one that's tried. Miller and Levine have tried, and others have tried. We talked about our textbook that we looked at last week. What's amazing to me, and this was brought out very, very well by Behe. I like this. Because when you read Richard Dawkins' book, what's the popularity of Richard Dawkins' books? He makes evolution seem easy. Of course it could occur. It's just so easy. He's deceiving us. It's not easy. You think it would be easy for that clotting cascade to have occurred evolutionary? It'd be impossible. But Darwin and people like, Dawkins and people like Dawkins, they want to make evolution seem easy so that it can occur. Very simplistic. It's just a breeze. And we know that's just crazy. Biochemistry was not a part of evolutionary synthesis. When Darwin was coming up with this theory, they didn't know biochemistry. And yet it must account for molecular structures in life, and it simply does not. Silliness popped up by such nonsense as Richard Goldsmith's hopeful monster. That was a desperate plea in the 1940s because he knew he couldn't explain how evolution occurred. So now you got a frog hopping out of a pond and becoming a bird. I mean, that's the hopeful monster concept. And then later on in the 70s, 60s and 70s, Stephen Gould and Niles Eldredge has punctuated equilibrium. We got to get something to punctuate it because we know all this slow stuff won't ever work evolutionary-wise. I thought this was interesting. Professor of biology at the University of Massachusetts, not a small university, a very renowned university, Lynn Margulis. She predicts history will judge neo-Darwinism as a minor 20th century religious sect within the sprawling religious persuasion of Anglo-Saxon biology. That is a good way of saying basically it's nonsense. And it just floors me why we are considered backward. When to me, as she points out, it should be and I don't know her religious background. I don't know where I know she's a biologist, and she may have, she may hold to some evolutionary concepts, but she certainly doesn't hold to neo-Darwinism, does she? So Dr. Margulis has challenged evolutionists to give just one example of the unambiguous formation of a single new species formed by the accumulation of mutations, and to date her challenge has gone unmet. Oh, that was quick. I apologize for that, but I wanted to get through. You got it in your handout. Any questions? There'll be a test on this cascade next week, by the way. No, I'm just joking. But this is simplified. This is not the whole cascade. I want you to know that. And there's still parts of the clotting mechanism they don't fully understand. But it's just, and you could go on and on. And like I say, you could look at the flagella, you could look at the cilia, you could look at the bombardier beetle that they show, that he talks about. And there's just umpteen number of these kind of processes that are going on in our body simultaneously while we sit here, and we're supposed to believe that there was no intelligent design in this. I just can't believe it for a second.